Have you ever wondered how our body get rid of damaged or unwanted cells without causing chaos? That's where apoptosis or programmed cell death comes in, an essential process that keeps us alive and healthy. Apoptosis is a process of programmed cell death. Cell death can be fundamentally divided into accidental cell death and programmed cell death. Programmed cell death is a genetically controlled cell intrinsic suicide process that eliminates specific cells. It is essential for controlling cell number, removing unwanted diseases or damaged cells, and maintaining the cellular homeostasis. Apoptosis and programmed cell death are often used synonymously. Although apoptosis is the most common form of programmed cell death, there is also non-apoptotic programmed cell death, such as autophagy and regulated necrosis. Autophagy is an intracellular degradation process of cytoplasmic component through the formation of autophagosomes. The autophagosomes fuse with lysosomes, the resulting compartment called autolysosomes, and they cause enzymatic breakdown of cytoplasmic components. The demise of cells by apoptosis is marked by a well-defined sequence of morphological changes. Apoptosis exhibits cell shrinkage, chromatin condensation, which is also known as pycnosis, nuclear fragmentation, and plasma membrane blebbing, accumulating with the formation of apparently intact small vesicles. These are also known as apoptotic bodies that are efficiently taken up by neighboring cells with phagocytic activity and degraded with lysosomes. Importantly, the intracellular constituents are not released into extracellular fluids, where they would probably have deleterious effects on neighboring cells, but instead are phagocytosed by neighboring cells. Chemical changes in the surface of apoptotic cells or bodies allow the surrounding cell or macrophages to recognize and engulf them. An especially important change occurs in the plasma membrane of apoptotic cells. The negatively charged phospholipid phosphatidyl-serine is normally exclusively located in the inner leaflet of the lipid bilayer of the plasma membrane, but it flips to the other leaflet in apoptotic cell, where it can serve as a marker of these cells. There is essentially no inflammatory reaction associated with the process of apoptosis nor with the removal of apoptotic cells, because A. Apoptotic cells do not release their cellular constituents into the surrounding interstitial tissue. B. They are quickly phagocytosed by surrounding cells or neighboring cells. And third, the engulfing cells do not produce any inflammatory cytokines. Why apoptosis is so important? It plays a crucial role in embryonic development, immunity, preventing disease. Let's take an example. It helps shape our fingers and toes during embryonic development by removing unnecessary cells. In frogs, you might have seen these webbed feet and hands. Those are the same webs present in the embryonic development of fetus. So apoptosis helps and remove these. It's also vital for removing cells that may become cancerous or cells that have been infected by a virus. Without apoptosis, we would have an accumulation of damaged cells, which could lead to diseases like cancerous, autoimmune disorders, and many more. Now let's break down the two main pathways of apoptosis, the intrinsic and the extrinsic pathway. Both lead to cell death, but they're triggered by different signals and different proteins are involved. Let's talk about intrinsic apoptotic pathway. Apoptosis is induced not only by exogenous signals reacting with death receptors, but also by intracellular death signal released from the mitochondria. Various cellular stresses such as DNA damage, hypoxia, and growth factors deprivation lead to release of cytochrome C from the mitochondria and activates the intrinsic or mitochondrial apoptotic pathway. This type of induced cell death has therefore been called apoptosis by neglect as counterpart to receptor-mediated apoptosis by instruction. Let's talk about mechanism of intrinsic apoptotic pathway. Let's say the cellular stress is DNA damage. There are certain signaling molecules that are found inside the cells which sense this DNA damage, and these proteins are like ATM, CHK. These are signaling molecules which binds to the damaged DNA. Once they sense and bind to the DNA damage, they activate P53. An activated P53 is very dangerous protein because P53 can turn on multiple proteins and other mediators will ultimately guide the cell through apoptosis. P53 will not allow this cell to pass to the next stage or phase of the cell cycle. It will halt the cell cycle for this cell. So now the P53 will start recruiting factors or proteins. One of such proteins is Bax or Bac protein that creates pores in the mitochondria. Bax creating pore in the mitochondria will allow a very important component, which is cytochrome C, to be released from mitochondria to the cytosol of the cell. Cytochrome C is released in the cytosol, which act as a death signal because this should not happen. Cytochrome C should present inside the mitochondria. Once it's coming out to the cytosol, the cytochrome C will pair with another protein called APAF1, which is apoptotic protease activating factor 1. Cytochrome C and APAF1 comes together and it will catalyze a very important activity, and they will activate a set of protein cascade reaction which will ultimately turn into a nuclease activation. So cytochrome C and APAF1 will go and activate caspase 9. Previously the caspase was present as pro-caspase. So once caspase 9 was active, it would go and it will further activate pro-caspase 3 into caspase 3. So now caspase 3 once active, it is going to further activate nuclease enzyme. Now nuclease enzyme will start nuclease activity in the cell and eventually leading to apoptosis. That's all for intrinsic apoptotic pathway. Now let's talk about extrinsic pathway. Unlike intrinsic pathway, which is triggered by internal stress, the extrinsic pathway is activated by signals from outside the cell. These signals come from molecule known as death ligands. Cell have many receptors, some for growth responses, some for survival signals, and other for death signals. One of the key receptors involved in the extrinsic pathway is Fas receptor or fatty acid synthase receptor, which binds to a specific signaling molecule known as Fas ligand or Fas-L, which is fatty acid synthase ligand. When a cell undergoes DNA damage or is infected, it responds by producing Fas ligand. Once Fas ligand binds to the Fas receptor, it activates the death domain inside the cell. This activation is autocatalytic and initiates a death-induced signaling cascade, which is DSC. In intrinsic pathway, caspase 9 play a key role, but in extrinsic pathway, caspase A takes on this function. The inactive procaspase A is activated by death domain receptor, converting it into active caspase 8. Caspase 8 not only plays a vital role in extrinsic pathway but also facilitate crosstalk with intrinsic pathway. In the extrinsic pathway, caspase 8 can activate caspase 3 from its inactive form, procaspase 3. Once activated, caspase 3 degrades an inhibitor protein that normally suppresses nuclease enzyme. These enzymes, when free, enter the nucleus and breaks down the nucleic acids, including chromosomes and DNA. This ultimately leads to cell death, which is apoptosis. And this is how the extrinsic pathway works. The importance of balance in apoptosis. Both intrinsic and extrinsic pathway leads to apoptosis. But the body needs a balance. Too little apoptosis and cells that should die may stick around and become cancerous. Too much apoptosis and healthy cells are destroyed, which can contribute to diseases like Alzheimer's. So that was a look at apoptosis and how it's essential for health and balancing in our bodies. Whether it is from internal signals in the intrinsic pathway or external signals in extrinsic pathway, apoptosis is a key to maintaining cellular health. That's all for this video. Thank you.